Okay, so this chapter 16 is discussing the control of gene expression in prokaryotic cells. The expression of genes in bacteria is often regulated through operons, which are groups of genes that are transcribed as one unit. So they produce a mRNA that is a single strand of mRNA, but ultimately that single strand of mRNA will produce multiple proteins. So what you're looking at here is a picture of E. coli, which is a common bacteria that is found in the intestinal tracts of mammals. Now, another reason why this is important is because E. coli was used to help identify the trip operon, which is an operon that produces three enzymes that all work together to synthesize the amino acid tryptophan. All right, so let's talk about genes, and in this chapter, and specifically this section, we're going to talk about genes and regulatory elements. We're going to talk about levels of gene regulation and DNA binding proteins. Now, all of these things are important and play a significant role when we start talking about how genes are regulated, and this requires one that you can consider or distinguish between DNA sequences that are transcribed and DNA sequences that regulate the expression of other sequences. Okay, so this really incorporates or allows you to think about structural genes. These are going to be those genes that encode proteins, and regulatory genes. These are genes which can be either RNA or protein that interact with DNA sequences and affect transcription and or translation. Okay, so as I stated here, structural genes are going to be the genes that encode proteins that are used in either metabolism or biosynthesis, or they play a structural role in the cell. So those are going to be structural genes. Regulatory genes are going to be either RNA or protein, code for either RNA or protein, and they interact with those DNA sequences that ultimately affect the rate of transcription or translation of those sequences. In many cases, the products involved in the regulatory genes are DNA binding proteins, or they can be RNA molecules that can bind to the DNA and alter the expression of those genes, ultimately by blocking the proteins from being able to move past or pass forward from that region of the DNA. And then you have regulatory elements, okay? Now, with regulatory elements, they affect the expression of DNA sequences when they are physically linked to them. Okay, so these would be common in both bacteria and eukaryotic cells, and a lot of the regulation in these both organisms, whether it's prokaryotic or eukaryotic cells, they take place through the activity or the actual action of proteins that are produced by regulatory genes. So regulatory elements can be produced by regulatory genes, and once these genes are produced, they will recognize and bind to the regulatory elements. Okay, so in some examples of that in bacteria, you have these gene regulation. In bacteria, you have gene regulation, and it helps to maintain what we call internal flexibility. So this is in instances where you would turn genes on or off. Consider it like a flashlight, a light switch, So if you go into a room, if the room is dark, you hit a switch to turn the light on, and you hit that same switch to turn it off. You see this similar concept, although it's simplistic, right, or superficial. It helps you understand how gene regulation works in regulatory elements. Their elements and their presence will actually bind to a specific area in the DNA and cause the turning on and off of genes. And a lot of times their physical presence actually acts as a blocking agent to prevent the machinery from actually binding to the DNA and carrying out transcription. A lot of times these regulatory mechanisms are influenced by environmental changes. So when a certain substance is present in the environment, it causes it to turn off, and if certain substances are absent in the environment, it causes it to turn down. And we'll talk about some of these examples, like the lac operon for lactose, or tryptophan. Okay. In multicellular organisms, you have gene regulation that also helps bring about cellular differentiation. And even with cellular differentiation, you have the presence of certain proteins in the environment that induce a change to occur. So this brings us to how these regulatory elements are altered, okay, or modified. You can have constitutive expression. This is where it's continuously expressed under normal conditions. And this would be an example of something that is needed, right? So it's always going to be expressed unless something is present to turn it off. And you can have positive control of that element or negative control. Again, keep in mind, we're talking about an operon. So we're talking about a cohort of genes that are linked together by a single promoter. Okay. And so whether those genes are expressed are determined by regulatory elements. Okay. If it has a positive control, then something has to stimulate that gene expression to occur. And if it has a negative control, something has to inhibit gene expression from occurring. So let's look at a physical example or visual example of that. When we talk about gene expression, there are different levels and mechanisms by which it can be controlled. You can control gene expression on the DNA level by the condensing of the DNA. So as you see here, it can be compacted or condensed. And if DNA is highly condensed, that means that the machinery cannot get access to the DNA to actually carry out that process of using it as a template. You can also have it relaxed. And when DNA is relaxed, that means it has the ability to be accessible. And so you can have transcription or the presence of the DNA being used as a template to make a transcript. You also have the rate during transcription or pre-mRNA and processed mRNA. So in these different areas, these processes can actually alter whether transcription or gene expression occurs or not. And then also through the modification of proteins, whether a protein is folded in an inactive or an active state will also regulate gene expression. So there's multiple ways in which gene expression can be regulated. And I want you to pay attention to this diagram here, because you're going to see when we start talking about these different forms of gene regulation through, let's say, operons, how these different levels of control play a role. All right, so let's talk about DNA binding proteins. Okay, these are proteins that have the ability to bind to DNA and cause a change, conformational change, thereby altering whether the DNA is accessible or not accessible, and also ultimately causing a chemical change in the DNA. Right? Domains typically or tend to be 60 to 90 amino acids in length, and they're responsible for binding to the DNA. How they bind is by forming a reaction, in this case, a hydrogen bond with DNA. And that binding allows for this domain to cause or allow for regulation, the modulation of accessibility of these proteins and their ability to bind to DNA. You also have something called motifs. Motifs are the structural type modifications or areas in the DNA that allow for further binding and modifications. For an example, you have in DNA major and minor grooves, and there are certain motifs that allow for proteins to bind and fit into that major and minor groove region. And these oftentimes are distinct proteins. Oops. All right, so here are some examples of these different types of motifs that we were talking about. So you have helix turn helix, okay, as you see here. And you can see here, this is the DNA binding helix. There's the turn, and then you have another helical structure there. You also have zinc fingers. Zinc fingers are the structural areas where you have a turn, the helix turn, but you have a zinc molecule that is bound in that region, and you often associate zinc fingers with DNA binding. A lot of transcription factors have zinc finger domains, okay, and this allows them to bind to DNA in a more efficient manner and allows them to be transcription factors. And then you also have leucine zippers, okay, and this is where you have two turns, helical turns, and the parallel binding, or the parallel nature of this domain allows for this interaction, this chemical interaction, to allow them to form this zipper-like structure. All right, so here you'll see a table to help you further understand some of those motifs we were talking about that allow for the various gene regulation or binding to DNA. And you'll see here, for example, as I told you with the zinc finger, we see this oftentimes in eukaryotic regulation and other proteins, but these typically tend to be transcription factors. And these transcription factors, have zinc at the base of that loop, and they allows them to bind to DNA in the major grooves of DNA. So this would be a very helpful study tool for you during the process of studying. So let's get into section 2, where we are going to talk about the operons, and how transcription is regulated by operons. So we have to talk about the operon structure, because the operon structure, again, has a promoter region, or leads with a promoter region, and has additional sequences that follow. The additional sequences that follow, and sometimes they occur in front of the operon or the promoter region, these additional sequences can determine whether you have positive or negative control. Whether it is turned on, or turned off. And a lot of times it is regulatory proteins that bind to these regions to determine the efficacy of these operons. So we're going to talk about the lac operon and the chip operon, both in E. coli. And we'll talk about the factors that control or regulate these operons. All right, so what is an operon? As I stated earlier, an operon is a sequence of DNA that has a promoter region and additional sequences that control transcription, in addition to structural genes. So you have an operator, that is sequences that help to control transcription, and then you have structural genes. Those are going to be the genes that follow the promoter region, and they oftentimes can code for other enzymes, And then you have a regulator gene. This is where you have a sequence of DNA that encodes a product that affects the operator. It's not a part of the operon, but it regulates the operon. It determines whether, i.e. regulator, whether it is expressed or not expressed, and sometimes how much it is expressed. So keep in mind, the regulator gene is a sequence that produces a product that determines or alters, i.e. regulates, the operon. And it is separate from the operon. So let's look at a diagram of that. So you see here, we have just a general example or view of an operon. So here is the operon right here. Here's your promoter region, and what overlaps from the promoter region is the operator. Then you have your structural genes that follow the promoter region. Notice here we have another promoter region and the regulator, a gene that codes for the regulator. The regulator, this sequence here, once it goes through transcription and translation, produces a protein, which is the regulator protein. And this regulator protein binds to the operator and controls or regulates the operon. Remember that, travel your mind back to that slide previously where I showed the levels of control. This is the example of the levels of different types of control. Now, the importance of the operon is these two sequences here, which are bound by RNA polymerase initially, determine whether these subsequent genes are expressed, gene A, B, and C. If they're allowed to be expressed, i.e. RNA polymerase can bind to the promoter region and carry out the expression of these genes through transcription and translation, then those 3 genes produce proteins or enzymes that can then act on other pathways. Okay, so you can see how a lot of these this operon regulates and can have a downstream effect on other processes in the body, or in this case, in bacteria, based off of whether the operon and these structural genes are allowed to be expressed or not. So, now that we have a basic understanding of operons, let's talk about the control of these operons, whether it's a positive control or negative control. Another term or way of looking at positive versus negative is induced versus repressed. Okay, if it is an induced operon, that means transcription is usually off and something needs to induce it to turn it on. Okay, so in a normal state, it's going to be in an off state. Okay, a repressible operon in a normal state is always on, and something has to be induced to turn it off. So there has to be something to repress the transcription. Okay, so in an operon that is normally off, an inducer has to turn it on. In an instance where a operon is normally on, something has to be induced. Something has to repress the expression. Okay, so with the negative inducible operon, the control at the operator site is negative. Okay, hence the molecule that is binding to the operator, which is inhibiting transcription. Okay, so there's a molecule, a protein that is bound to the operator, and it is blocking transcription, hence why it is off. Okay, and so when something is bound to the operator, it is in an off state. Something has to be induced to turn it on, and the induction is usually going to interact with the molecule that is binding to the operator to prevent it from binding there. So it's a compensatory mechanism that blocks it from being bound to the operator. Okay, and that molecule is the inducer. The inducer is a small molecule that turns on the transcription by binding to the molecule that is bound in the operon, the operator. So here you have the instance where there's no inducer present, okay? And in that case, the operator is bound to the regulator protein is bound to the operator and no transcription is occurring. This is in a normal state. It's in a off state. And so you need the presence of an inducer, okay? The inducer, if present, will bind to the regulator protein, preventing it from being bound to the operator, thereby allowing for RNA polymerase to come in and carry out gene expression. Okay, again, remember back to the hierarchy of the levels of expression, and some of them are environmentally regulated, hence the presence of this inducer in the environment. Okay, the presence of this inducer in the environment will bind to the regulator protein, allowing for expression to occur. All right, let's talk about the negative repressible operon, or the negative repressor. Okay, in this case, the control at the operator, is negative. That means there's nothing bound to the operator, so the operon is turned on. Okay, it is turned on in a normal state, and so there has to be what we call a co-repressor, which is a small molecule that will bind to the repressor and make it capable, as you see here, of binding or from being unbound to the operator. So in a normal state, the operon is turned on. There's no co-repressor there. So the co-repressor is not allowing for the repressor to bind to the operator. Therefore, the genes here, the structural genes, are expressed, and therefore they're able to then act on other pathways to carry out functions. However, if the co-repressor is present, the co-repressor will bind to the repressor, which allows it to bind to the operator, thereby turning transcription off. Okay, so again, you have some operons, that are inducible, and then some operons that are repressible. So let's talk about the positive and negative control. Okay, positive control of a operon is where you have the regulatory protein that is involved and is acting as an activator. So it will bind to the DNA and stimulate transcription. However, in a negative inducible operon, okay, so let's specifically talk about the lac operon. The lac operon is an operon that is associated with what we call lactose, okay? Lactose. And the lac operon is a negative inducible operon, and it is involved in lactose metabolism. Specifically, it regulates the lac operon. The inducer in the lac operon is a molecule called allolactose, okay? Allolactose. And so it is another form of lactose. Hence, remember again, go back to the levels of expression, the controls of expression. If lactose is present, okay, then you can have the modification of this operon, okay? The three structural components here, you have the LAC-I, which is the repressor encoding gene. LAC-P is the operon promoter, and LAC-O is the operon operator, okay? The structural genes are part of the LAC operon are LAC-Z, Y, and A. And each of these three genes encode for enzymes that are involved in the synthesis or the breakdown metabolism of lactose, okay? The repression of the LAC operon never honestly completely shuts down, but it goes into a very low state. And this is also associated with glucose expression. Okay? Glucose expression. What do you mean? Well, what is lactose? It's a major carbohydrate that's found in milk. Okay? And it consists of about 2 6-carbon sugars that are linked together. I want you to keep in mind that glucose is the preferred source of sugar or nutrients for the cell. But when there is no glucose, the cell has to find a way in which it can provide and produce the molecules it needs to survive. And so in the case here, if lactose is present, while it's not the preferred, the cell is going to utilize the lac operon to break down lactose into glucose, into a usable form or the preferred form to produce energy. So here you have lactose, okay, that is outside of the cell and it's transported into the cell. Once it's transported into the cell, you have beta-galactosidase, one of the genes, that structural genes from the lac operon, that breaks down lactose into galactose and glucose. Okay, hence why the lac operon is never fully turned off, but it's present in very low amounts, so that when lactose is present, you have small amounts of these enzymes that can readily break it down to produce glucose. Okay, and so what happens here, you have lactose that can also, small amounts of lactose that can also be acted upon or converted by beta-galactosidase to convert it to what we call allolactose, okay? And converting allolactose, remember what is stated about allolactose, okay? Allolactose is a inducer for the operon, okay? So these are actually needed. All right, so let's look at the actual operon itself, the lac operon, okay? So here you have the lac operon in section A, and this is what happens in the lac operon when lactose is absent. So when lactose is absent, the regulator protein that is encoded for by the regulator, LAC-I, is bound to the operator, and no transcription of the operon is occurring. And RNA polymerase cannot bind to the promoter region and carry out the transcription of these genes, these structural genes. So when lactose is absent, you have the operon that is turned off. When lactose is present, lactose can bind to the regulator protein, which then prevents it from being bound to the operator, as you see here, therefore turning the operon on. So when lactose is present, the operon is on. When lactose is absent, the lac operon is turned off. Okay, just to kind of show you how it looks, here's the lac promoter. There's your consensus sequences that we talked about. Here's the lac repressor. okay? And it's bound to the operon as the operator, okay? And it can begin to carry out the synthesis of those structural genes. So let's talk about the different forms of control, okay? With the lac operon, let me see. So with the lac operon, you have what we call catabolite repression, okay? And this is associated with the presence of glucose, okay? Glucose acts as a positive control. It determines the function of the operon. So when glucose is available, it represses the metabolite of other sugars. So another way of saying that simplistically is when glucose is present, there's no need for an alternative form of energy. So the lac operon is turned off because lactose is not present, and the preferred form of energy is glucose. Okay, so that is the positive control of the lac operon in terms of glucose. Okay. Now, what the more details of the positive control of the lac operon is that you have what we call a catabolite activator protein, or CAP. Okay. Cyclic AMP binds to CAP, and together you have this CAP cyclic AMP complex that binds further upstream from the lac gene promoter. Okay, and this is also helpful in the control of the lac operon. Cyclic AMP concentration is actually inversely proportional to the level of glucose. Okay, so cyclic AMP is the level or the expression concentration of cyclic AMP is inversely proportional to the presence of, or the availability of, glucose. So let's look at that here. Okay, this CAP complex has to bind with cyclic AMP before binding to the promoter of the lac operon. Okay, so when you have the binding of the cyclic AMP CAP to the promoter, it activates transcription by facilitating the binding of RNA polymerase. Okay, so the levels of cyclic AMP are related or inverse related to glucose. When there's low glucose, it stimulates high cyclic AMP. And when you have high glucose, it stimulates low cyclic AMP. So there's an inverse relationship between the levels of glucose and cyclic AMP, and thereby regulating the lac operon. So here, when glucose is low, you have cyclic AMP levels that are high, that are readily available to form this complex, thereby increasing the efficiency of the polymerase binding, saying, hey, we need an alternate form of energy, produce lactose, break it down because we prefer glucose. Okay, so break down the lactose so that we can get glucose. However, when glucose is high, this operon is not needed, so you don't have high levels of cyclic AMP. Therefore, the rate of transcription is significantly reduced. So you have little to no level of transcription of the lac operon. So this is a form of positive control in that it helps to further enhance the rate of transcription via the presence of this molecule cyclic AMP and glucose. Okay, so this is just visually showing you the actual binding complex of cyclic AMP and CAP and its ability to help make the binding of DNA, RNA polymerase to DNA, much more efficient. All right, so let's go into the trip operon. The trip operon is an operon that is involved in the synthesis of tryptophan, the amino acid tryptophan, and this is specifically was found in E. coli. Okay, now what happens? You have an operon that has a promoter region and an operator that is bound by a regulator protein. Okay, and again, this is a negative repressible operon, and the presence or the sequencing or the synthesis, of these structural genes will determine whether tryptophan is expressed or not. okay, and primarily the ability to convert other molecules to produce tryptophan. There are 5 structural genes in this operon as opposed to 3 in the lac operon, and it's TRP A, B, C, and D, and all these enzymes together to help convert the chorismate to tryptophan. So let's look at this operon, okay? Again, when tryptophan levels are low, okay, you have the operon, the regulator, or the repressor, I should say, that is unable to bind to the operator. Therefore, the CHIP operon, is turned on, and so you have now the production of tryptophan. Okay, so when there's a need for tryptophan, the operator is on, the operon is on. Therefore, the regulator or the repressor is not bound to the operator. When tryptophan levels are high, there is no need to produce more tryptophan, so the operon is turned off. Therefore, the repressor is bound to the operator, and you have the co-repressor that is bound to the repressor, keeping it in that state. Okay. Lastly, attenuation. So what is attenuation? It is a mechanism for reducing expression, okay, of the CHIP operon when levels of tryptophan are high. Okay. However, rather than just blocking initiation of transcription, attenuation prevents completion of transcription. So when you have high levels of tryptophan, attenuation causes RNA polymerase to stop prematurely when it's transcribing the CHIP operon. Okay. So ultimately what happens is you have this short, stubby piece of mRNA that is made, and it doesn't encode tryptophan.